Welcome back to botany class. I hope that you have completed all the definitions related to the topic genetics. Let me draw your attention towards today's learning objectives. You are going to focus reciprocal cross, analytical and empirical approach, test cross, back cross, molecular explanation and dihybrid cross. Let us begin with reciprocal cross. What's meant by reciprocal cross? In monohybrid cross, you learned that the tall plants, that is capital T is crossed with dwarf plant small t. So, it is tall and this is dwarf. In F1 generation, it gives capital T and small t. In one experiment, this capital T, the tall is taken as female and the dwarf is taken as male, or this is reversed. When the dwarf is taken as female and this tall is taken as male, the result is same. So, this is called reciprocal cross. So, in this F1 generation, this is the parent, this is F1 offspring. In F1, all plants are tall. So, Mendel concluded that in reciprocal cross, this reciprocal cross is not sex dependent. So, Mendel's monohybrid cross is not sex dependent. What is the analytical and empirical approach? In this experiment analytically, in F1 generation, this dwarf character, the dwarf is hidden, it is not expressed. In monohybrid cross, it is a single trait which contains two alleles. This is one allele, this is another. Single contrasting character. The character is height of the plant, where in F1 generation, when two contrasting characters, one single trait with two alleles crossed in F1 generation, it produces all tall plants, and F2 generation, it gives the ratio 3:1. When these plants are self-crossed. So, in the first generation, the dwarf is hidden. Here it is not expressed. The gene is there, but it expresses the character tall. Dominant characters expressed. This dwarf is not disappear, but it is hidden or masked, and it is reappear in the F2 generation. So, in F2 generation, it gives the ratio 3:1. So, this capital T small t crossed with capital T small t. So, it gives capital T, here capital T small t, here capital T small t, here small t. This is homozygous tall, these two are heterozygous tall, and this is dwarf, only one dwarf. Now, this dwarf is expressed. In F2 generation, the dwarf is reappeared among the 1/4. It reappears. So, Mendel's calculations, the analytical ratios are accurate. So, he was the first person to give the quantitative explanations, quantitative studies of this type of experiments. So, he was called as father of genetics, and his experiments are meticulous, carefully observed. All the calculations, all the ratios are carefully observed. Empirical approaches, due to this, the observable characters, observed nature, empirical approach. Here it shows that there is a single trait or factor or a particulate. Under particulate theory, there is a particulate unit which carries the trait which transmits generation after generation. So, this is called as gene. Now, this is named as gene. So, these genes are carrying the characters. These characters are expressed generation passing, or it is transferred generation after generation. Next topic is test cross. What is meant by test cross? Test cross is a cross between unknown parent with the recessive character. So, recessive in under monohybrid cross, the recessive character is dwarf. So, test cross is any one of the parental form, the unknown form. So, it may be the capital T or it is capital T small t, either with capital T or with capital T small t. These two are possible gametes in the tall plants, which is crossed with recessive parent, that is small t. So, unknown parent is crossed with recessive parent is called as test cross. So, it shows two different types, homozygous recessive and heterozygous recessive. So, what is the reason of this test cross, or what is the use of this test cross? Mendel selected a contrasting character in monohybrid cross. So, F1 generation it produces all tall plants, and in F2 generation Mendel cross this dwarf into dwarf. So, F2 dwarf is crossed with F2 dwarf, it gives dwarf in capital T in F3 generation as well as F4 generation. So, he found out the recessive trait is small t or the dwarf is small t. But what is the reason behind this? Homozygous tall and heterozygous tall. So, F3 and F4 generation it produces dwarf. So, he conclude that the dwarf gene is small t, dwarf trait. But how to predict homozygous tall and heterozygous tall? Among these 3:1 ratio, this is homozygous tall, these two are heterozygous tall. So, how to predict the genotype is homotypic or heterotypic? So that he gives this ratio, he gives the unknown genotype is crossed with this recessive. So, he get the ratio 1:1 ratio. Let us calculate it with analytical type of using checkerboard. Here capital T, when you make a cross between this capital T into small t, it gives capital T small t, capital T small t, capital T small t, capital T small t. All are heterozygous. When homozygous tall is undergo test cross with the recessive parent in F1 generation, it gives all tall plants. But heterozygous, this, let us find the ratio of this. Capital T small t and small t, it gives the ratio capital T small t, capital T small t and small t. So, these two are small t and these two capital T small t. So, the ratio is 1:1 ratio. So, heterozygous it produces 1:1 ratio. Homozygous tall it gives all tall plants. This is to predict. So, test cross is the cross is used to predict the homozygosity and heterozygosity of tall plants. Then what is meant by back cross? So, test cross, this is the cross between unknown parent with the recessive recessive, pure breeding recessive parent. There are two different types, homozygous, heterozygous. Undergo homozygous stall with recessive parent, heterozygous stall with recessive parent. In back cross, the hybrid is crossed with any one of the parent. Here which is the hybrid under monohybrid, these all crosses based on monohybrid cross. So this is the F1 hybrid. The hybrid capital T small t is crossed with any one of the parent, that is either capital T or this hybrid is crossed with capital T small t. The parent, either it is a parent, this small t. So the back cross is the cross between the hybrid with any one of the parent, F1 hybrid. This is the F1 hybrid, capital T small t, F1 hybrid is crossed with any one of the parent. This is the parent, either the homozygous stall or the homozygous recessive. So it shows the ratio when you make a cross between these two back cross. It is also two different types. They are dominant and recessive. So it is dominant and recessive. This character, this is crossed with either any one of the parent. So it may be tall. It is dominant character. Tall is dominant. This is dwarf, recessive character. So it is classified into two types, dominant back cross and recessive, dominant and recessive. Find out the ratio. Here this capital T and small t and capital T. So it gives capital T, small t, here capital T, small t. So it also gives the ratio 1:1. Then find out the analytical ratio using checkerboard. This gives heterozygous or the hybrid. Hybrid is crossed with this capital T, small t and this small t. So it gives capital T, small t, capital T, small t. So it also gives the ratio 1:1 ratio. So in back cross it gives 1:1 ratio. So these all crosses based on this monohybrid cross. So hope that you understand the concept of this monohybrid. Under this, what are the related ratios or what are the related crosses? Then what is the molecular explanation of this tall and dwarf plants? You know that Mendel selected 7 contrasting characters. One of the contrasting character is height of the plant or length of the plant. What's the reason? The pea plant, the garden pea, which produces tall and dwarf. So molecular analysis, molecular basis, it's interesting. Here when they find out, here in the pea plants, it produces a precursor. The cells of pea plants which produces a precursor molecule that is formed of gibberellin. Gibberellin producing precursor molecule. So this gibberellin producing precursor molecule, the pea plant, the tall plants, this tall plants contains an enzyme. So gibberellin, the activated form of gibberellin is GA1. So the cells of pea plants contains the precursor molecule, gibberellin activating molecule called as GA1. And the tall plants which encodes it contains a gene or allele capital L small e. This is the allele which is present in the tall plants. When this allele is present, it activates, it encodes a protein. It is a functioning enzyme. which activate this gibberellic acid 1 GA1. So, when it is activating this, the normal functioning of formation of gibberellin, this gibberellin it functioning due to this activity of GA1, it produces the tall character, it shows tall. So, the tall allele is either capital LE or capital LE or capital L small e and small l small e. So, this all alleles which gives tall. Then what is the reason of this dwarf character? It gives small l, small e, small l, small e. Recessive form of this allele, it gives dwarf because it shows that inactive nature of this GA1. So, enzyme which do not contain this capital L small e, instead it contains the recessive form. So, it is the inactive nature of this GA1. This is the molecular explanation of tall and dwarf plants. Now, let us move to dihybrid cross. In monohybrid cross, there is a single trait, monohybrid, single trait is taken. A single trait which contains two different alleles. Height of the plant, the character is height of the plant. But in dihybrid cross, there are two contrasting characters are selected. In monohybrid cross, there is only one character, single trait contains height of the plant, tall and dwarf. But dihybrid cross, di which means two, dihybrid. As the name indicates, di, two contrasting characters are selected. So, this is based on the law of independent assortment. So, first under monohybrid cross you will learn two laws, law of dominance and law of segregation. Here based on this dihybrid cross there is a law of independent assortment. How it is carrying out? So, dihybrid cross, the characters are, what is meant by dihybrid cross? Two contrasting characters are crossed and make a hybrid. So, it is the dihybrid inheritance which the characters are selected as round yellow, round, the shape, there are two characters, the character is shape of the seed, and color of cotyledon. What are the characters? The shape of the seed and color of cotyledon. So, Mendel selected two contrasting characters which contains the shape. The shape of the seed is round and wrinkled. Round and wrinkled. The color of the cotyledon is yellow and green. So, it shows two contrasting characters. The two contrasting characters are round yellow. So, characters are round yellow and wrinkled green. So, the characters are taken like this: capital R, capital R, capital Y, make a cross with small r, small y, small y. When you write this capital letters and small letters, write it very clearly because you have to find the ratio under this. 9:3:3:1 is the ratio under dihybrid cross. If you write this gametes, alleles and the gametes taken out correct, the proportions you can get it correct. Or else if the letters, if you confused with the capital Y with the small y, you confused when you are writing it in the checkerboard. So write it clearly. Capital R, this capital R represent round and capital Y represent yellow cotyledon. Round, this is round, this is wrinkled. This is yellow, this capital Y represent yellow dominant character, this is the recessive character. So this is the round yellow. This seed, you draw a diagram like this and it shows wrinkled green. It shows the character round yellow. This is wrinkled green. This is dominant over this. The capital R represent the shape of the seed round. Small r represent the shape of the seed wrinkled, and capital Y represent the color of the cotyledon yellow. Yellow is dominant over green. This Y, small y represent the green color. So first you understand clearly what are the contrasting characters. The contrasting characters in dihybrid crosses are two contrasting characters. Here dihybrid which contains two contrasting characters, round yellow, wrinkled green. So this is taken as a parent. The parents are round yellow and wrinkled green. Then what are the possible gametes? These are alleles. What are the possible gametes are present here? Here capital R, capital Y, small r, small y. So here in the book the diagrams are given in the form of chromosomes, which shows the chromosomes which shows the genes. So when you draw the diagram, you draw it clearly. Which connects here capital R and capital Y, capital Y. So it shows this yellow, capital R, round shape. This is the chromosome. The genes are located within the chromosome. Here yellow. Likewise here. Here small r, wrinkled, and this small y is the green color. Such a way all these characters you draw the chromosomes clearly. Here capital R, so 1 from here capital R, this one, and capital Y, with this. Here small r which contains and small y, this. So cchromosomes carrying genes. So this is the parent, these are the gametes. Gametes are capital R, capital Y, small r, small y. When a cross between this round yellow and a wrinkled green, this one, in F1 generation, it produces capital R, small r, all possible combinations. The gametes are combined together and expressed in the F1 generation. So the possible gamete is capital R, capital Y, small y. Capital R, capital Y, small y. In this dihybrid cross, this at the time of gamete formation, so in it shows the independent assortment, law of independent assortment. What is meant by law of independent assortment? When two contrasting characters are combined at the time of gamete formation, these characters are segregated independently. It assort independently. So, at the time when all possible gametes are combined together, at the time of separation, the gametes are assort independently. All combinations are here, but these all gametes, this is F1 generation. This F1 generation contains all the combinations of genes. 4 possible combinations are present. Here, in this F1 generation is self-crossed with capital R, small r, capital Y, small y. So, in hybridization experiment, F1 is a hybrid. So, it leads to evolution, a hybrid, and this hybrid is crossed with another hybrid. So, what are the possible gametes here? This capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. There are 4 combinations are possible: capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Likewise, here also capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These 4 combinations are possible. So, it assort independently. If all these characters are combined together, it assort independently, is called as law of independent assortment. It assort independently the genes. Now, when you take this ratio, you will get dihybrid cross. When you make a cross between this self, this hybrid with hybrid, the same F1 into F1, F1 into F1, self crossed, you get 16 possible gametes, where it shows round, yellow round, yellow round, yellow ringled, ringled, round yellow, yellow ringled, then ringled yellow, yellow, and ringled green. So, these are the 4 possible gametes from this ratio. So, when you take this gametes, one take it as male and this set take it as female, you get the ratio 9:3:3:1. Let us check the ratio, how it appears. Make it in the form of checkerboard. There are 16 possible gametes. Here one set take it as male, another set here take it as female. So, write it here: capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Here this side capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now, fill this. Here capital R, capital R, capital Y, capital Y. Here capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Here capital R, capital R, capital Y, small y, capital R, this and this, small y, this and this, capital R, small r, capital Y, small y, capital R, small r, small y, small y. And here capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y. Then last, capital R, capital Y, small y, capital R, small y, small y. Here, small r, capital Y, small y, and small r, small y, small y. Now, take out the ratio. The expected ratio here is 9 is to 3, 9 is to 3 is to 3 is to 1. So, here the capital R and capital Y, this shows capital R and capital Y shows that round yellow, capital R, capital Y, yellow, round green, this one wrinkled yellow, this is wrinkled green. So, all these 4 possible gametes are available. Here capital R and capital Y, if it contains capital R and capital Y, that is round yellow. Here round yellow, round yellow, round yellow, round yellow. Here capital R but small y is there. So, round green. Here round yellow, round green. Here round yellow, round yellow. This is wrinkled yellow. Smaller represent wrinkled. Wrinkled yellow, wrinkled green, capital Y is there. So, this is wrinkled yellow, Y. Here round yellow, round green, wrinkled yellow, and the last one it shows pure recessive, that is wrinkled green. Check the ratio. Round yellow, it's 9. 1, 2, 3, 4, 5, 6, 7, 8 and 9 is to round green. 1, 2 and 3 is to wrinkled yellow. 1, 2, 3 is to 1 wrinkled green. So, it shows that 9 is to 3 is to 3 is to 1 ratio. So, this cross is called as dihybrid cross. So, what is the difference between monohybrid and dihybrid cross? In monohybrid cross, a single trait is selected, but in dihybrid cross, 2 different characters are selected. 2 different characters, 2 contrasting characters. So, it based on the independent assortment, law of independent assortment. Now, what is meant by dihybrid test cross? In dihybrid test cross, it gives the ratio 1 is to 1 is to 1 is to 1. So, dihybrid test cross, the ratio it gives 1 is to 1 is to 1 is to 1. So, what is dihybrid test cross? The hybrid is crossed with recessive parent. This is the hybrid. F1 hybrid is crossed with the recessive parent. So, capital R, capital Y, small y into small r, small y. These are the parent. Take out the gametes from here. Here capital R, capital Y, capital R, small y. Small r, capital Y, small r, small y. 4 combinations of characters. Here only one, small r and small y. So, take it in the form of checkerboard. 1, 2, 3, 4. Here capital R, capital Y, capital R, small y. Small r, capital Y, small r, small y. For better illustration, we are taking it in the form of checkerboard. Then small r, small y. It is easy to take out the probabilities. Now, this capital R, small r, capital Y, small y, capital R, small y, small y, small r, capital Y, small y, and small r, small y, small y. So, here it gives the ratio 1:1:1:1. So, how it is? This is round yellow, capital R, capital. So, it is round yellow, this is round green, this is wrinkled yellow and this is wrinkled green. So, each one combinations it gives 1:1 ratio. So, 1:1:1:1. One round yellow, one round green, one wrinkled yellow and one wrinkled green. This cross is called as dihybrid test cross. I hope that you understand very clear. Note today's assignment. So, first question, reciprocal cross. It is a 3-mark question. Analytical and empirical approach also 3-mark question. Test cross and back cross. Either it may ask a 5-mark or 3-mark because 2 types are there, each contains. And molecular explanation 5-mark question. Dihybrid cross, very important, related to independent assortment. Thank you.